Okay, so welcome back to this next video in which we are discussing the mammalian circadian clock. Okay, so we've now discussed how glutamate is going to activate the expression of period and cryptochrome within suprachiasmatic nuclei neurons. Okay, we now want to look at this other signaling molecule released by the axon terminals of the photoreceptive ganglion cells, which is PACAP, pituitary adenylate cyclase activating protein. Okay, and this is going to activate a receptor on the membrane of the suprachiasmatic nuclei neuron known as Pac1, PAC1. Okay, right. So when the PAC1 receptor is activated, what will happen is you'll get a change in conformation in the G protein coupled receptor here, which will be translated down to a change in conformation of the free intracellular loops. Okay, so these free intracellular loops will change conformation and they'll make available a binding site for the alpha S subunit of a heterotrimeric G protein. Okay, so let's draw this. So what I'm going to do is I'm going to spin the G protein coupled receptor around 180 degrees, and all this is artistic license. Okay, I just want the carboxylic acid terminus to be out of the way basically when I'm actually drawing the picture. So here is the carboxylic acid terminus over here. And now what's going to happen is to these free intracellular loops, which I'll colour in orange again, so these free loops here, we're going to get the binding of the alpha S subunit of our heterotrimeric G protein. So in it comes, okay, like so. And it must be associated with a beta and gamma subunit in order to be able to bind to the intracellular loops. So the beta-gamma complex doesn't actually bind itself to the G protein coupled receptor, but without the beta-gamma complex bound to the alpha subunit, which is in this case is an alpha S subunit, the alpha subunit can't bind to the intracellular loops of the receptor. Okay, so this portion is essential. So a lone alpha subunit isn't going to be able to bind to the intracellular loops of the G protein coupled receptor. Okay, so there's the beta subunit in blue. Here is the gamma subunit in green. Okay, and the alpha subunit we'll have in red here. Okay, right. So the alpha subunit within the heterotrimeric G protein binds to the free intracellular loops of our Pac1 receptor. What then happens is the binding of this alpha S subunit to those free intracellular loops is going to catalyze the alpha S subunit releasing its guanosine diphosphate that is bound to it, and instead binding guanosine triphosphate from the cytoplasm. Now, the reason that this requires catalysis is that temporarily, when you remove the guanosine diphosphate from this binding site here, you will temporarily have nothing bound in there. Now, the state of having nothing bound in there is very high energy usually. So if we draw one of these little energy diagrams, okay, we'll have the state of having guanosine diphosphate bound is like so. Then the state of having GTP bound is around the same energy. Okay, but to get from there to there, you have to temporarily go through the state of having nothing bound, and this is a high energy state, so you require quite a large activation energy usually to give up your guanosine diphosphate, which is why alpha S subunits don't usually spontaneously activate. Okay, the effect of binding the alpha S subunit to the G protein coupled receptor is to lower the energy hugely of that state where you have nothing bound, so that you can more easily give up the guanosine diphosphate, go temporarily into this nothing state, and then rebind guanosine triphosphate. Okay, right. And basically, in common sense language, what it comes down to is that the binding of the intracellular loops to the alpha S subunit makes it easier for the alpha S subunit to give up its guanosine diphosphate. That's the common sense argument for what I've said there. Okay, right. Now, when the alpha S subunit gives up its guanosine diphosphate and instead binds to the GTP, what then happens is the beta-gamma complex cleaves off. It can't bind to the alpha subunit once the alpha subunit is in the on state, so the beta-gamma complex cleaves off. Now, once the beta-gamma complex is gone, the alpha subunit can't bind to the G protein coupled receptor anymore. Remember, I told you that for the alpha subunit to be able to bind to the intracellular loops of the G protein coupled receptor, it had to have the beta-gamma complex bound. It's now lost the beta-gamma complex because it's turned into the on state, so it too is going to fall off the intracellular loops of the G protein coupled receptor. And what this means is we're going to get the alpha S subunit now in the on state going off in the cytoplasm, well, going off in the underneath the inner leaflet of the phospholipid bilayer on its own little adventure, and the beta-gamma complex will also go off on its own little adventure. Okay, so let's now see what signaling pathways the alpha S GTP subunit is going to lead to. Okay, so basically the alpha S GTP subunit is going to go and activate adenylyl cyclase enzymes. So this is the next important topic for us. We need to discuss adenylyl cyclase enzymes. So here is our alpha S subunit with the GTP bound. Now, we don't really care about the beta-gamma complex anymore. We just care about the alpha S subunit with the GTP bound. Okay, so adenylyl cyclase enzymes then. There are 10 adenylyl cyclase enzymes found in humans. Okay, 9 of them are what are known as membrane-bound adenylyl cyclases. Okay, so 9 are membrane-bound, whilst the final one is what's known as the soluble adenylyl cyclase. Okay, so the final one is called SAC, and what this stands for is the little s stands for soluble, and then the AC stands for adenylyl cyclase. Now, this soluble adenylyl cyclase looks nothing like the others, okay, and it's the one that we're not going to talk about because it's very scarcely found in human cells. It's found a lot in the testes, but apart from that, it's not really that present within normal cells. Okay, so we're going to study the 9 membrane-bound adenylyl cyclases. So cross this one out, we don't care. Okay, so the 9 membrane-bound adenylyl cyclases are called adenylyl cyclase 1 all the way to adenylyl cyclase 9. And what's the Roman numerals for adenylyl cyclase 9? There we go, that's 9. Okay, right. And they all have pretty much the same structure, which I'll draw for you now. Okay, so all of these membrane-bound adenylyl cyclases, they are a single polypeptide. Okay, so the whole enzyme is made up from one polypeptide. Now, again, let this represent the phospholipid bilayer, the inner leaflet and the outer leaflet. So this is the cytoplasm, this is the extracellular fluid. So, the amino terminus of the polypeptide is going to be in the cytoplasm. Okay, then you're going to have a membrane-spanning alpha helix, and it's going to be clustered together with 5 more membrane-spanning alpha helices. Okay, so overall you're going to have this cluster of 6 membrane-spanning alpha helices all together, and this is known as the transmembrane domain 1, or TMD1. So this stands for transmembrane, that's the TM. Okay, T for trans, M for membrane. Then the D stands for domain. Okay, so transmembrane domain 1, TMD1. Then you have a loop between TMD1 and then another cluster of 6 membrane-spanning alpha helices over here, known as TMD2. Okay, so this is the transmembrane domain 2. And then you have a cytoplasmic tail here, culminating in the carboxylic acid terminus here. Okay, right. So let's just highlight up the different portions. So here in vivid purple are the 6 membrane-spanning alpha helices of TMD1, and here are the 6 membrane-spanning alpha helices of TMD2. Now, the purpose of TMD1 and TMD2 are just to hold the adenylyl cyclase enzyme in the phospholipid bilayer. They are not the catalytic portions of the enzyme. The catalytic portions of the enzyme are made up by this loop in between TMD1 and TMD2, which is known as the C1 domain, okay, and the cytoplasmic tail here, which is known as C2 domain, okay. Right, now C1 domain and the C2 domain are further divided up into two portions, okay, so C1 is divided into two portions. One portion that I'm now colouring in green here, which is called C1a, okay, and then another portion which I'll colour in orange here, which is known as C1b, okay. Similarly, C2 is divided up into two separate domains called C2a here, and then C2b here. So C2a I'll colour in green again, and then C2b I'll colour in red here. Okay, so those are the divisions of these different domains. Now, the active enzyme is made up by a dimerization of the C1a domain with the C2a domain. So if you join C1a to C2a, you then get the active adenylyl cyclase enzyme, which will be capable of catalyzing the cyclase reaction. Okay, so what I have drawn is an inactive adenylyl cyclase enzyme because the C1a domain and the C2a domain are not dimerized together. So let me now show you an active adenylyl cyclase enzyme. Okay, right, so once again I'll draw the phospholipid bilayer here, and here still is the amino terminus here. Then we've got TMD1, which is this cluster of 6 membrane-spanning alpha helices, and once again I'll colour code these in purple here, like so. Then you have this loop between the final membrane-spanning alpha helix of TMD1 and then the first membrane-spanning alpha helix of TMD2. So we then have the 6 membrane-spanning alpha helices of TMD2, which again I'll colour in purple here. Okay, so this is called TMD1 and TMD2, of course. And now what we want to show is the C2a domain dimerized with the C1a domain. So I'll use a little bit of artistic license again here and stretch C2 out rather a lot from this picture here. But I can't show them dimerized otherwise. So let's say this now is C2a here. Okay, so in green, this is C2a, and this is C1a, also in green. Okay, and then the other domains, let's show them. We then have C1b here, so this is C1b, this is C1a, this is C2a, and then finally we'll add on C2b in red down here. Okay, and once C1a and C2a have dimerized together, that then produces the active enzyme which is capable of catalyzing the cyclase reaction. Okay, so what I want to now show you is what the cyclase reaction is, and then we'll talk about how the alpha S GTP subunits are going to activate adenylyl cyclase enzymes, i.e. promote the changing of these inactive adenylyl cyclase enzymes into these active adenylyl cyclase enzymes. And I want to stress that what we are discussing here goes for all 9 of these adenylyl cyclase isoforms here. They all look like this and they all activate in the same way, and they're all activated by alpha S GTP subunits. Okay, right, so let's now talk about the cyclase reaction. Okay, so the cyclase reaction involves taking adenosine triphosphate, ATP, and converting it into cyclic AMP and pyrophosphate. So let's discuss this. So ATP stands for adenosine triphosphate. So let's draw a little cartoon of adenosine triphosphate. So to do this, we need to know what adenosine is. Now, adenosine is the name for adenine bound to a ribose sugar. So I'll start by drawing a ribose sugar here. So it's a pentameric structure with a 5th carbon coming up here. And I'm not drawing a formula for it, I'm just drawing a cartoon for it. So this blue pentagon here with the 5th carbon coming off up here, this represents the ribose sugar. Okay, and then coming off the 1st carbon of the ribose sugar down here, because remember this is an oxygen atom. Okay, you then have the organic base adenine. Okay, and I'll represent adenine just as a rectangle with adenine written in it, like so. Okay, and I'll colour that in green here. Right, so that now is adenosine. Adenosine means adenine bound to ribose. So the combination of these two is then called adenosine. Okay, right. So how do we convert adenosine into adenosine triphosphate? Well, we stick 3 phosphate groups coming off the alcohol group of the 5th carbon up here. Okay, so here are 3 phosphate groups up here, and this converts adenosine from adenosine to adenosine triphosphate, just meaning adenosine with 3 phosphate groups, or ATP. Now, this is the substrate for our active adenylyl cyclase enzyme. What we're going to do is we're going to convert adenosine triphosphate into cyclic adenosine monophosphate, usually abbreviated to CAMP. Okay, so cyclic adenosine and then monophosphate, okay, which is usually abbreviated to CAMP. C for cyclic, A for adenosine, and then MP for monophosphate. And the other product that you're going to produce is something called pyrophosphate. Now, pyrophosphate is basically 2 phosphate groups bound together. Okay, so let me show this. So just 2 little phosphate groups bound together, and it's usually abbreviated to PP for pyro and then phosphate, and then you put a little I there, and that little I is in the same spirit as when denoting a phosphate group you denote it PI. It means inorganic. Okay, so what you're going to do is you're going to cut this bond between this first phosphate here and this second phosphate here. And by the way, the phosphates of adenosine triphosphate are all given names. The first phosphate is called the alpha phosphate, the second phosphate is called the beta phosphate, and the third phosphate is called the gamma phosphate. So we're going to cut the bond between the alpha phosphate and the beta phosphate in adenosine triphosphate. That will give us a pyrophosphate molecule, which is the beta phosphate and the gamma phosphate coming off there. Okay, and now what we've got left there would be adenosine monophosphate. Adenosine, adenine plus ribose, and then monophosphate with a single phosphate group. We are going to convert this into cyclic adenosine monophosphate by binding this alpha phosphate group to the alcohol group that comes off this 3rd carbon down here. Okay, so what's that going to create? Well, that's going to create something known as cyclic adenosine monophosphate. So you've still got your ribose with your adenine here, and I'll just demote it to being represented as a rectangle with A written in it. Then you've got your 5th carbon coming off up here, then a phosphate group coming off that 5th carbon. But the phosphate group is now linked down to the 3rd carbon of the ribose sugar, like so. Okay, so in blue there we have our ribose sugar, and in green we have our adenine. So this now is cyclic adenosine monophosphate. It's adenosine monophosphate with this extra cycle that's formed here within the structure. Okay, so it's cyclic adenosine monophosphate. And that conversion of adenosine triphosphate to cyclic adenosine monophosphate and pyrophosphate is known as the cyclase reaction. Okay, right. So the final thing to say about adenylyl cyclase enzymes is how the alpha S GTP subunits activate these adenylyl cyclase enzymes. Basically, alpha S GTP subunits bind to the C1 domain and they bind to the C2 domain, and they pull them close together, and that promotes the dimerization of C1a with C2a. So I'll say that again. The alpha S GTP subunit is going to come along and it's going to bind to some point on the C1 domain. It's then going to have another site which is going to bind to some point on the C2 domain, and that ends up bringing the C1 domain here close to the C2 domain, and that promotes the dimerization of C1a with C2a, and therefore promotes the enzyme changing from being in the inactive form to being in the active form. Okay, so when PACAP binds to its receptor, the PAC1 receptor, and activates alpha S subunits, what you're going to get is the activation of adenylyl cyclase enzymes. Okay, and these will start converting ATP to cyclic AMP. Okay, right. In the next video, what we'll discuss is what the cyclic AMP is going to do, which is activate protein kinase A enzymes. And we'll then discuss how protein kinase A enzymes, just like calcium calmodulin-dependent kinase enzymes, can then phosphorylate serine 133 on Krebs binding protein. Oh, sorry, not on Krebs binding protein, on Krebs proteins. Okay, which then allows Krebs binding proteins to bind. So we'll do that in the next video.